Well, what happened to all the labs across the Gulf obviously was that people were moving along with their research programs in their labs, and then suddenly you have this huge catastrophe with loads and loads of oil out there, and everybody immediately switched. Either they were, we've talked to a number of scientists who continued doing what they've always done, but they had to do it in the context of the presence of oil and how that impacted the environment they were working in. But we're very interested in knowing with you, after doing the research you've done now for the last couple of years, what are your new research questions? Do you, anytime a scientist does research, everything you do raises a new question, and we're interested in knowing what yours are.
Okay, yeah, that's a good question. I think that's an important one in terms of people who've been working in and around the Mississippi, Louisiana coast and the Mississippi Delta. You can't look at it as being the same system you were working in pre-spill. So some of the work that we were doing, we had already been involved in some of the work with hypoxia, and that had a big impact on what we were doing even in 2011 with our cruises through the hypoxic region, and thinking about, what are the effects if we're adding more, or if this material is leaking out from the marshes or from sediments, and there's residual material out there. How do we factor that in to the 2011 interpretation of the 2011 hypoxia area and so on? And so that's been a difficult one to factor in, and it's one that we have addressed by adding some additional hydrocarbon types of analyses, which were not done before. We had this ongoing work addressing those questions with no oil, and now we have in these marshes, and it just so happens that Barry Bay, has some interesting areas where we have mixtures of both, and so we are now involved in actually coring some of those areas that I described earlier that have been oiled, and the ones that have not been oiled, whether they have mangroves mixed in or not. And so we're now, just as we're doing in the water, we're actually looking down core, and we have some students focusing on oil deposits and seeing if that oil has the chemical signature that matches with the spill. And so, because you do have accumulations, the sediments are essentially a repository or a history of what's happened over many years. And so, and there are natural oils that come from plants decomposing.
At the same time as you're trying to answer one set of questions, you're always making observations that bring about new questions. And so certainly there are new questions. And for us, one of the big sweeping questions that we have when it comes to planktonic systems in the Gulf of Mexico, a very productive system, is how hard do you have to push the system to actually get it to tip over to something that can't return or does not return to the state that it was in before? In other words, how hard do you have to push perturbation in this system to get it to go to what we call an alternate stable state? It doesn't mean it's an alternate bad state. It just means it's something different. And so for us here, that's now a driving question, is how hard do you have to push the system to get it to go into another alternate stable state?
It's been interesting because of the one example that's brought up constantly from Exxon Valdez of the herring fishery in Prince William Sound. Our counterpart organism is menhaden. And nothing's probably more important in the Gulf of Mexico in the food chain for the higher animals, at least, than menhaden. But they are particle feeders, and they're indiscriminate. And so they, of all the species of fish that we're concerned with, here was a keystone species that might have been exposed to floating particles of oil and things of that nature. And I think that's one of the things that we're kind of watching, I think, has to be a concern, because everything eats menhaden, including dolphins.
Well,
so, I think
you have different, not all fish are the same. So you have Some fish are big fish, big groupers and snappers, things that live for many years, and they can probably get by. They can move, they can swim around, they can get by. What we need to look at are the little fish that make up the majority of the food, right? So you're talking about things like menhaden and anchovies and herrings, things like that. So those fish, we call them forage fish. We really don't know much about their populations as it is. We don't know how much is out there, how much the system relies on them to transfer energy from plankton up to larger fish. So there's a tremendous absence of data on stocks of forage fish. So we need to look, I think at that level. And how do you do that retrospectively if you don't have the data from before? Well, we can use models. We can rely a lot on models, and I think there should be a large push on modeling efforts of fisheries production from this ecosystem perspective. And that's it's a direction that we're generally moving in terms of management of our fisheries is to take this ecosystem look. And so there is attention that's being paid to forage fisheries, things like menhaden, what their role is, because they're an exploited fishery as well. And so we need to know about them anyway. So let's take advantage of this event and push forward, this agenda to know more about what our forage fish populations are like here in the Gulf.
Oil is organic matter. Organic matter is food for some organism. I mean, we may not eat oil directly, but certainly there are microorganisms that can eat it and can eat the gas, the natural gas that was present also. What does that do? That, of course, can ultimately deplete oxygen because these microorganisms utilize oxygen just like we do. And in the depletion of oxygen, then, we can affect the chemistry of the metals and affect their partitioning between the dissolved phase and the particulate phase, meaning that we can affect their bioavailability or toxicity. So these sorts of secondary questions become much more important as we proceed through this event.
This really has actually started to change the direction of our laboratory in a significant way. We're not going to do a complete 180 from our direction. However, this definitely gives us an advance in knowledge that we didn't have before this spill. A lot of researchers out there look at these natural seeps of methane and even some crystalline forms of methane, an ice-like structure on the seafloor called a methane clathrate hydrate, that potentially can release a large quantity of this gas. With it being a potent greenhouse gas, can potentially propagate to the atmosphere and influence climate. That still is very much true, and a lot of it depends on the depth at which these releases occur. Deep water releases are a little bit more challenging to make it up to the atmosphere than a shallow water release. And certainly areas of the world's oceans may not be as populated by these bacteria that are accustomed to consuming this methane. But before this spill, what my focus was primarily on was trying to measure, trying to put a quantitative number on how quickly is methane being released from these seeps and potentially making it up then to the atmosphere to influence the greenhouse gas budget and climate change in a natural way. My research direction has taken a slight tangent to that now, and instead of trying to just ultimately quantify the absolute number that's coming out of the ocean due to these natural seeps, due to these methane clathrate hydrates, but to instead now to quantify the capacity, the kinetics of these bacteria that are capable of consuming this methane. If we have this sink, this removal of this greenhouse gas, what are the limitations of it? Are there chemical limitations? Are there physical limitations, such as temperature, that can limit how quickly it can consume methane? This event in the Gulf showed us that these bacteria can go a lot faster than kind of the conventional wisdom was for how quickly they could respire this gas. If they went as fast as we saw in the Gulf, how fast really can they go? What are their limitations? Getting kind of a fundamental biochemical understanding of their kinetics is really where our laboratory is going at this stage.
Wow, that's fascinating. The applications that can come out of this are really, really going to be interesting.
You know, going forward, part of the lesson of the spill from 2010 was that we don't understand some of the critical functions and processes that occur in the Gulf of Mexico, despite many years of research by scientists in the region. It historically has been relatively underfunded compared to other regions in the U.S., other coastal regions, and we think that now that the spotlight has been cast on the Gulf, that there will be more substantial funding opportunities to start to address some of these data gaps and to help us better understand how these different habitats relate to each other, how they relate to commercial activities like fishing, oil extraction, dredging, and some of those other things that go on in the Gulf at the same time and same space, same location as some of these important habitats that we'd like to preserve so that we can continue to enjoy the benefits that they provide.
In terms of the natural methane research that's out there, while we have shed a significant amount of light on how that plays out in a global context and interacts with the atmosphere, or doesn't interact with the atmosphere, we've by far not killed this area of science at all, and not said that this is something that's maybe not relevant at all for climate change, and if you're interested in studying it, you're only interested in studying it in an esoteric sense. That's nowhere near where we're at. There's still many areas where methane is seeping and venting out of the seafloor and going directly to the atmosphere. And in those locations, normally the water depth is a lot less than what we had here in the Gulf. But even in these deep water releases, we're talking in the Gulf, the BP oil spill, of having a jet release. That's very different than having a slow bubble coming out of the seafloor. And the kinetics on how that dissolves or doesn't dissolve changes substantially. And even if we were to say that a released methane source was to be completely absorbed in the ocean and completely respired by bacteria in the ocean, the end product of that respiration is carbon dioxide. Carbon dioxide, as we know, is also a greenhouse gas. And more carbon dioxide in the ocean also contributes to the process of ocean acidification. So both of those avenues, as well as the diversity of oceanographic environments, shows us that our knowledge of this system, this methane system, this massive greenhouse gas reservoir of methane that is in ocean sediments, it's far from complete.